Hi, my name is Mia Luth, and the book that my group chose to do was The Immortal Life of Henrietta Lacks. Henrietta Lacks is an African American woman who unfairly had her cells taken from her without her permission or knowledge. These cells, called the HeLa cells, would later become some of the most important cells in scientific and health discovery. This book was written by Rebecca Skloot. Rebecca Skloot was a scientific journalist who had a lot of interest in the HeLa cells and wanted to find out more where they came from. She contacted Henrietta Lacks' family and found out that Henrietta had passed away many years ago. With the help of Henrietta's daughter, Deborah, she wrote this book that would later become a #1 New York Times bestseller. It would also become a very successful movie. This book told a lot of people about Henrietta that they had never known before. Rebecca also sat down with Henrietta's children and grandchildren and explained what the HeLa cells were, the benefits, and how they worked. Before this time, Henrietta's family had no idea what the HeLa cells were. It actually took them 30 years from when the cells were taken to even find out that these cells still existed and were immortal. Even then, it would take another 30 years before Rebecca found them and explained to them what these cells did. After this book came out, the family finally received some recognition, which we will talk about later. To understand this a little more, we need to understand Henrietta. Henrietta grew up under the poverty line with her grandfather and her older cousin. She worked on a tobacco farm until she was 21, and she married her older cousin. They moved to Maryland, where she began to work in a steel factory. There, Henrietta faced a lot of troubles. Not only was working in a steel factory hard, but she had 5 children, one who was mentally ill and would later die in the sane asylum. She also faced abuse from one of her cousins, so she did not have a very happy life. Despite this, she maintained positivity and tried to live her life as best as she could. About 4.5 months after giving birth to her last child, she found out that she had cancer. She started to go to Johns Hopkins for treatment. It was the only place that would help an African American woman at that time in the area. While she was under treatment, without her knowledge, they took some of her cells from her cervix. These cells would become the HeLa cells. They never told her, they never asked permission, and about a year later, she died of cancer without knowing. Henrietta was buried for 60 years before receiving a proper burial with a tombstone that was donated by a college professor. After this book came out, her family started to finally receive some of the recognition that she deserves. HeLa cells are very important. Just a quick summary of some of the things they've done is they've helped us with HIV, they helped us create a polio vaccine, helped us with cancer research, just understanding cells in general, and they've even been to space. One of my group mates will talk a little bit more about HeLa cells and their uses later on. Basically, though, the big issue here is the moral issue. Despite all that these cells have done and the fact that they are all over the world, Henrietta's family has barely received any recognition or any profits from this. Most of her family still lives under the poverty line. These cells are being traded, and they're probably thousands, if not millions, all over the world. Her family will never know how many of their mother's cells still exist or what they were being used for. They did receive some things after this book came out that made it a little bit better. They received recognition from the National Cancer Society and the Smithsonian. Their mother also received an honorary degree.
Hi, I'm Veronica. So in 1951, Henrietta Lacks went to Johns Hopkins Hospital after finding a lump inside of her cervix. She had began to bleed and was in excruciating pain. She was examined by a gynecologist by the name of Howard Jones, who oversaw what was referred to as the Black Unit. He confirmed that there was a lump and completed a routine biopsy. The results came back positive for epidermoid carcinoma of the cervix, stage 1. This portion relates to the mutation in cancer portion of class, where her cells in her cervix began to mutate into cancerous cells. This will be discussed later by Jimmy. While Jones was treating Henrietta for cancer by packing her cervix full of radium and sealing it so it didn't contaminate anywhere else in her body, he removed cells from her cervix without her knowledge or permission and sent them to the lab of George Guy. George Guy was a scientist who ran a lab in the janitor's closet of the same hospital with his wife Margaret. Guy's research methods would help us to understand mitosis and the cell division process by utilizing PCR, or polymerase chain reaction. This is the rapid production of one sequence of DNA into millions of copies. PCR is DNA testing in a test tube. These tubes contain DNA polymerase enzymes, supply of 4 types of nucleotides, and 2 types of short laboratory-made printers that are complementary to opposite ends of the target sequence. This testing produced more than thousands to millions of replications in a short amount of time. Guy's methods of PCR originally began by using a machine called a roller drum. This machine ran 1 cycle every hour. The latest model of this machine is called a thermal cycler, which controls temperatures and can spin thousands of times in a minute. These cells were produced so rapidly that Guy shipped them off to hundreds of researchers around the world to use for their own research. This was a complicated process that took years to perfect, as every time they would ship them out, pressure from the airplane or temperature change would cause the death of these cells. As stated before, these researchers utilized the rapid reproduction of HeLa cells to make many vaccinations. Henrietta cells saved over 300,000 white people that were infected by polio. They've helped with birth control, cloning of the first human cell, the discovery of the 46 chromosomes, and what can happen with an extra chromosome, like with Down syndrome. They also recently assisted in discovering in vitro fertilization. It's been almost 70 years since Henrietta Lacks was diagnosed with her cells, and her cells have been immortalized. They're said to live forever and have lived longer than she ever did.
My name is Jimmy, and I'm going to discuss what the HeLa discovery means to the science world. So, first of all, scientists have been trying to keep human cells alive in culture for decades, but they all eventually died. Henrietta Lacks, this patient who ended up going to Johns Hopkins due to cervical cancer, the doctors found her cancer cells to be part of an immortal cell line. So this means that due to a mutation in her cells could keep dividing without ever dying, and they reproduce an entire generation every 24 hours, and it never stopped. So her cells were the first immortal human cells ever grown in a laboratory. It was a great achievement for science, obviously. And after she died, scientists built a massive factory for her cells, with the main goal being to stop polio. The staff of the factory grew to 35 scientists and technicians, and 20,000 tubes of HeLa got produced, which is about 6 trillion cells every week. And this is just of Henrietta Lacks' cells, nobody else's. And so this all started from the first vial that the Dr. Gay had sent Cher in their first shipping experiment. And with her cells, they proved that the vaccine was effective to stop polio. So her cells really started the virology field, which is the study of viruses, like research into the genes and those, like the genes that cause cancer and those that suppress it. So her cells helped develop drugs for treating herpes, leukemia, influenza, hemophilia, and Parkinson's disease. And they've been used to study lactose digestion, sexual transmitted diseases, appendicitis, human longevity, mosquito mating, and the negative cellular effects of working in sewers. So their chromosomes and studies have been studied with such detail and precision that scientists know their every quirk. It's kind of like, think about like guinea pigs and mice. Henrietta cells had become the standard laboratory workhorse. And the early cell culture and cloning technology developed using HeLa helped lead to many advances that required the ability to grow single cells in culture, like isolating stem cells, cloning whole animals, and in vitro fertilization, also known as test tube babies. So, a quote I would like to share from the book was, by someone who said that Henrietta's cells have been living outside her body far longer than they ever lived inside it. If we went into almost any cell culture lab in the world and opened its freezers, we'd probably find millions, if not billions, of Henrietta's cells in small vials on ice.